Well, we're about to finish the semester, and teachers be like the purple minion, but I'm looking forward to being hula minion. Okay, we're going to go over chapter 20, which is the gastrointestinal system. Now, I did some of this lecture already, but I'm starting at the beginning, and you can put me on triple time if you want to get to the part where you want to go over things. But looking at the overview of the gastrointestinal system, we consume nutrients, we consume potential energy in food and vitamins and minerals, and they enter our mouth. And that is the beginning of the gastrointestinal tract or the digestive tract. And these nutrients are too large to consume. So as you can see with the little mouth, they get broken down by teeth mechanically. And then salivary amylase will also enter the mouth through the saliva. So salivary amylase and saliva enters, and that starts the breakdown of carbohydrates. So this is chemical equation for carbohydrates. The food bolus is swallowed and sent down the esophagus to the stomach. And the stomach is where proteins start to get digested. So proteins start chemical digestion. Here you have carbohydrates start chemical digestion in the mouth. And we'll talk about stomach. You have hydrochloric acid that will unfold or denature those proteins, and then proteases, which are enzymes that start breaking down the proteins into smaller chunks. What is created in the stomach is mixed by the 3 layers of smooth muscle into a substance called chyme, and chyme is then sent to the small intestines. Now, the small intestines is where fats start to break down. With the help of bile from the liver, gallbladder, which is an accessory gland. So bile enters, and pancreatic enzymes and buffers from the pancreas enter the small intestines as well. And this is where the rest of chemical digestion occurs. And everything's broken down into the smallest monomers, and then you have absorption into the interstitial fluid and then into the bloodstream, right? So this is blood. So you have secretion from the accessory glands and chemical digestion, and then you have absorption once those molecules are all broken down. Now, what does not get absorbed stays in the colon. So stuff not absorbed. And eventually it's compacted, and unabsorbed material is then defecated out. What you did absorb then goes into your bloodstream. Now remember, the mesenteric capillaries become mesenteric veins. And the mesenteric veins send nutrient-rich blood through the liver and then into general circulation, at which point the rest of the cells get to take what they need from what you absorbed. So the liver really does kind of filter this nutrient-rich blood that you just absorbed all these nutrients and maybe some not really desirable things, but all of it is going to filter through your liver cells or hepatocytes. And then that filtered blood is going to enter the inferior vena cava into the general circulation. And so it's important to kind of understand the overview. The gastrointestinal tract, it's a long hollow tube that changes shape between the esophagus. It just gets bigger to the stomach, gets a little smaller to small intestines, gets bigger with the large intestines, and then gets smaller for the rectum and anus. The accessory organs are the liver, gallbladder, pancreas, and salivary glands, and they are secreting things. And this is also showing you how the trachea is anterior to the esophagus. So up until the larynx, the digestive tract and respiratory tract share the pharynx. This kind of goes over from anatomy, all of the processes of the different parts of the digestive tract. And there is no homeostasis, meaning your digestive tract has evolved for maximal absorption of nutrients. And so your digestive tract is going to keep absorbing all the nutrients put into it. And if you're not using it at that point in time, it will be stored as a later source of energy. We call that fat in adipocytes or lipids. So whether it's too much, too many carbohydrates, too many proteins, too much fat, it all gets stored eventually in adipocytes or fat cells if your cells do not need those nutrients at that time. The gastrointestinal tract has it changes shape, but there are still the same basic mucosal lining. That's the most inner lining. And it's epithelium, really. And so that epithelium, you always have these epithelial cells right here that are absorbing, especially in the small intestines. And you have a lamina proper. It's kind of like a little connective tissue layer of mucus, and then a little muscle layer. And then you have the submucosa. Now the submucosa is where the blood supply is located. When you get to the submucosa, there is a little submucosal plexus. This is kind of the, what's called the brain of the gut. There is an independent little nervous system plexes. And so this is nervous tissue that will start the process of peristalsis and segmentation, the contraction of the smooth muscle. And that's what we call the enteric nervous system. It's controlling the smooth muscle. So all along the gastrointestinal tract, there's circular smooth muscle and longitudinal smooth muscle. And in the stomach, there is an additional third layer, which is oblique. So this kind of goes through the functions of the mucus. It helps move things along. It protects. So it's really important. The epithelium is simple columnar and mucus secreting cells. Then you have the loose connective tissue, and beneath it, you have a little muscle layer. You have lots of lymphatic cells throughout the whole gastrointestinal tract to kill off any pathogens that might be in the food that you consumed or what you drank. And so these enterocytes, these are the epithelial cells. They're responsible for absorbing or secreting. And they are the ultimate lining of the lumen, which is, lumen is the inside of the tract, right? So the inside of the tube, tract or tube. So study those, understand that. Go through the submucosal layer. This is where the large blood vessels and lymphatics exist for absorbing into the bloodstream. And then you have the enteric nervous system, the submucosal plexus. With the muscularis, you have smooth muscle, and you have the myenteric plexus. It's the second part of that little nervous system, or brain, if you will, that can initiate contraction of smooth muscle. On the outer layer, you have the serosa, if it's in the abdominal peritoneal cavity, and if it's above the diaphragm, it's just connective tissue. Now, again, the gastrointestinal tract in the small intestines is designed for maximal absorption of nutrients, and so because of this, it's evolved a very high surface area for maximal absorption. Right? So if you have a big surface area, you have lots of space to absorb everything across that epithelium. And so what that is you have these plicae. And so plicae are kind of these rolling hills. And so that right there increases the surface area because they're every so often. And then on the plicae, you have all these increased surface areas, and these are called villi. And so this is what the villi look like. It goes up, and this is probably small intestine. It's showing you intestinal crypts, but this is increasing the surface area. When you take a closer look at one villi, you're going to see that that one villi here, this is a villi here, has the epithelium with microvilli. And microvilli are these little extensions on the cell membrane. So these are extensions of the cell membrane. And so these are referred to as brush border. So microvilli are the brush border in the small intestine. And so these brush borders, so I'm coming up here to kind of show you, oops, sorry, that we're looking at a villi here, and this right here, this is the epithelium, and these are simple columnar with microvilli on the, what would be called the apical surface and facing the lumen. So this is the inside. And if I could only write, lumen is equal to the inside of small intestine tube. So the inside of the tube. So these microvilli face inside and they increase the surface area. So you can see this is increasing surface area. And because it kind of looks like a comb or brush, it's called a brush border. And in the small intestines, there are these little enzymes. They're located here on the brush border, and they're called brush border enzymes. And we'll talk about those when we talk about absorption in the small intestines. So that's the brush border. All right. So this increases surface area for maximal absorption. When there is food or substance inside the tube, it's going to stretch and it can activate the submucosal and myenteric plexes. And that will help control the enteric nervous system to move things along. The central nervous system also can control the enteric nervous system via the autonomic nervous system. So that would be parasympathetic activity. Hormones that are secreted by the endocrine cells in the stomach and small intestines. You have gastrin in the stomach and CCK, secretin, and GIP in the small intestines. This also helps contract smooth muscle, and also these hormones will increase the secretions from the accessory glands. So functional anatomy, you have the mouth that goes back to the oropharynx, and then you have the pharynx, laryngopharynx, and then it splits into the esophagus. You have salivary glands. You have two parotid salivary glands on the side, two submandibular and a sublingual. Again, salivary glands produce salivary amylase, and that starts the process of carbohydrate digestion. Esophagus, it's bigger. Stomach, the pancreas, by the way, is posterior to stomach. Gallbladder is found inside the liver, in between the lobes of the liver, I should say. The gallbladder stores bile, the liver creates bile, and it gets stored. So you can live without a gallbladder because the liver creates the bile. Then the stomach turns into the small intestines. There's the first portion, which is retroperitoneal. That's the duodenum or duodenum, which then comes back intraperitoneal and becomes the jejunum and the ileum, which then eventually opens up and becomes the colon. Colon has ascending hepatic flexure, transverse flexure, splenic flexure, descending sigmoid colon, and rectum. So going through the mouth, it'll talk about saliva and the properties of saliva. So take some time to go through this. salivary amylase, goes through the accessory glands that we've talked about, and we'll talk about these a little bit later. Saliva also has enzymes called lysosomes that break down bacteria. And so in addition to salivary amylase, there's a little antibacterial property to saliva as well. It also helps dissolve the chemicals in the food you ate to excite chemoreceptors in your mouth, which we call our taste buds. So you have to have saliva in order to truly taste the chemicals in your food. Saliva stimulation is activated by the parasympathetic nervous system. The sympathetic nervous system can activate protein-rich viscous saliva and inhibit watery saliva. That's more that when you have anything that activates your sympathetic nervous system, or you're just really nervous, you can get kind of thick saliva. That's not really helpful as much for tasting your food. Okay, so chewing, you guys can read through the chewing reflex. Normally jaw muscles are always contracted or activated, but when you put something in your mouth, it relaxes those muscles of mastication. So your food or your jaw opens up, and then you can chew that food by contracting your muscles of mastication. When you first swallow, that is volunteer, and it goes down in the pharynx, and as it gets to the laryngopharynx, hopefully the epiglottis covers over the trachea so you don't breathe in your food, and the bolus of food goes down the esophagus. There's an upper and a lower esophageal sphincter. They have to relax to allow the food into the esophagus and out of the esophagus into the stomach. This lower esophageal sphincter is associated with gastric reflex. So sometimes the lower esophageal sphincter, there's something pushing up, and it will push things through that lower esophageal sphincter back up into the esophagus. Anything that comes from the stomach back up towards the esophagus is reflux, specifically gastric reflex. Swallowing also initiates relaxation of the stomach muscle to accommodate the bolus of food that is going to be entering it. So in addition to relaxing the lower esophageal sphincter, the muscle of the stomach relaxes. So please read through these slides. Okay, so the stomach, it mechanically mixes what enters it. And there's some chemical digestion of proteins. Now, the electrical activity of the smooth muscle is excited by your autonomic nervous system, specifically the parasympathetic nervous system. It can be inhibited by sympathetic nervous system. But also, we'll talk about hormones, gastrin, that will activate the smooth muscle as well. Peristalsis, by the way, is the circular muscle contracting, longitudinal muscles relaxing and pushing things forward through tubes, whereas segmentation is alternating contractions between the types of smooth muscle, and that is for mixing, like in the stomach. In the stomach, as the food comes in, it enters the stomach, and the whole time this food is in the stomach, it is turned into a substance called chyme. Chyme consists of denatured protein, broken down carbohydrates, and fats. Now, to denature protein, remember the primary structure of a protein is a string of amino acids. And the secondary structure is an alpha helix or beta pleated sheet. And that's just a folding, a folding, let's just say a folding of amino acids, primary structure. And then there's going to be a third folding called the tertiary structure, and that has to do with the amino acid side groups. Those are the R groups. And denaturing is breaking down this tertiary structure, and it brings it more into a linear type of structure. And that's due to the hydrochloric acid. It's really acidic, and it makes those proteins just kind of fall apart. Then the Enzymes, a generic name for enzymes that break down proteins are proteases. Then the protease called pepsin, once the protein's denatured and kind of comes apart, the pepsin, the protease, can break the primary, secondary protein structure into smaller chunks. In addition, the stomach secretes, some of the cells of the stomach secrete intrinsic factor, which is necessary for the absorption of B12. If you lack intrinsic factor, you will end up with pernicious anemia. Remember, we need for healthy red blood cells to carry oxygen, adequate O2, you need hemoglobin. And for hemoglobin, you need vitamin B12, amino acids, and iron. So we need that vitamin B12 to be absorbed, and that is due to intrinsic factor. Now, once this chyme is all mixed up, it gets very liquidy, and it's going to be passed slowly into the duodenum or duodenum. And there's this pyloric sphincter, which is the muscle that relaxes to let chyme into small intestines or duodenum. So a couple of things: alcohol, aspirin, these things pass through the stomach mucosa and into the blood of the submucosal lining in the stomach. And so because of that, these substances that can pass through, lipid soluble substances that can pass through the stomach mucosa, are hard on the stomach lining. And so that's why a lot of alcohol and things like aspirin can give people stomach issues or gastritis, which is an irritation of the stomach lining. The stomach stores food. It expands as more food is added to it. And it contracts to mix things into chyme. As chyme is exiting the stomach into the duodenum or duodenum of the small intestines, it's called gastric emptying. The folds in the stomach to accommodate expanding when more food is put in there are called rugae. That's just the description of those folds. Now, waves of peristalsis, this contracting of the smooth muscle, helps mix the chyme and propel it back and forth towards the pyloric sphincter. And the volume or how much chyme that's in the stomach is going to determine the strength of the contraction of the smooth muscle of the stomach. And things that regulate this are the release of gastrin and CCK, secretin, and GIP. The stomach has a layer of bicarbonate-rich mucus to offset the pH of 2 to 3, which is extreme acid, right? Because of the hydrochloric acid. And so you have to have this bicarbonate-rich, which is a base, to offset that so it doesn't disrupt everything below that mucosal lining. Damaged epithelial cells are quickly shed and replaced because it's epithelium. And so this is why you can irritate your stomach lining or get gastritis. And if you have Tums or some kind of base to cut down on the acid, it'll allow those epithelial cells to regenerate. And so stomach surface or epithelium is turned over every 3 to 6 days. And so that's why Tums or Alka-Seltzer can be effective in helping kind of restore the epithelial cells and raise the pH so it's not damaging them too much. This really low pH, this acid, it activates pepsinogen, which is an inactive protease, to break down proteins. Proteases are enzymes. And that's just a generic name for enzymes that break down proteins. So hydrochloric acid activates pepsinogen, and hydrochloric acid activates pepsinogen and denatures protein and kills bacteria. And so hydrochloric acid is really important, but you don't want to have too much hydrochloric acid because it will disrupt the gastric mucosal barrier. So it does have bicarbonate in that mucosal barrier to help out. These right here are referred to as gastric pits. These pits go down, and there are cells all along the way. So the upper cells are called the mucosal and neck cells that also secrete mucus, and they're secreting that bicarbonate-rich mucosal lining. Deeper down into these gastric pits, you have chief cells, which secrete pepsinogen. And that is, again, the inactive protease. And then you have parietal cells, and parietal cells secrete hydrochloric acid and intrinsic factor to absorb vitamin B12 from small intestines. So that intrinsic factor gets mixed into the chyme, enters the small intestines, and helps with the absorption specifically of vitamin B12. So gastrin increases the force of the contraction. And so what's happening is the stomach's contracting, it's mixing back, it's mixing, then sloshing it kind of towards the pyloric sphincter, in which the sphincter relaxes a little bit and lets chyme enter. And so the chyme is basically entering slowly, a little bit at a time, into the duodenum. And that's really going to be dependent on how much food is in your stomach and the composition of the food. So this is supposed to be carbohydrates. It's supposed to be C6H12O6. If there's fats, because remember, fats don't start to break down until the small intestines. And so if there's fat and proteins mixed in with carbohydrates, it's going to stay in the stomach a little bit longer. It's going to take a little bit longer to empty at a slower rate into the duodenum, and it's going to then absorb into the bloodstream slower as well. So how long chyme stays in your stomach is really dependent on the composition of the food that you ate. So again, understand the cell types: mucosal, neck cells, parietal cells secrete hydrochloric acid. This is giving you what it does, right? It activates pepsinogen into pepsin, which is an active protease. Denatures proteins, so then the proteases can break them down into littler chunks. It can help break down some of the cellulose, just make it fall apart to get the nutrients out of complex carbohydrates, and can kill most bacteria ingested by food. In addition, it secretes intrinsic factor. So one is hydrochloric acid, two is intrinsic factor. Chief cells secrete pepsinogen, and pepsinogen, again, is an inactive protease. And in the presence of hydrochloric acid, it will then get converted to pepsin, which is an active protease. Enteroendocrine cells, and